All right, so we're going to start talking about Punnett squares, and this is something that I find some students know really well and others struggle with a little bit. So this is just a video that kind of walks you through the process, and definitely if you're having any trouble, you still need to see your teacher about how to do these anyway. So let's kind of get started on a couple of things. We have talked earlier about alleles and genes, and so just kind of for the purpose of this, we represent alleles when we do these Punnett squares with letters, A, B, C, D, so on and so forth. And so anytime we have a capital letter, we would indicate that being a dominant allele. And anytime you see a lowercase letter, it is indicating that that is showing a recessive allele. So in this case, I'm going to use letter T. The uppercase T is dominant for the dominant trait, which will be tall in what we're doing today, and the recessive trait, which is represented by lowercase t, will be short. Anytime, in order to make the organism and to show its genes, we must have a diploid cell, and to do that you must have two chromosomes paired up, and so we have to have two T's combined to make the trait, and one of those is coming from mom, one of those is coming from dad. So in this case, we could put the T's together in one of three combinations. You could have two uppercase T's, two lowercase t's, or you can have a mixture of upper and lowercase. Now, also we've got to know some terminology here. So we have the terms homozygous and heterozygous. If we say something is homozygous, both letters will be the same. So homozygous tall would be capital T, meaning that it's dominant tall, and both letters are the same. They're both uppercase. And then homozygous short would be two lowercase t's, meaning they're both the same, and they're showing that trait. The opposite of homozygous is heterozygous, and in that both letters would be different. So you would have an uppercase letter and a lowercase letter. In this case, I would say this as being heterozygous tall. Heterozygous meaning that both letters are different, and then tall meaning that it's showing the tall trait by having the dominant allele. Now, we're going to do Punnett squares, and basically a Punnett square is a diagram that shows all possible genetic outcomes. Now, the key word is possible. It's a prediction, so just because a Punnett square predicts it doesn't mean it's going to happen. But we use it to predict probabilities over a long term, and usually the probabilities are pretty close. A couple terms that are going to pop up with these Punnett squares that we haven't talked about yet, or briefly have, is the term phenotype. And a phenotype is an observable trait, so the phenotype we're going to use here today is tall and short. Those are phenotypes. The other term we're going to see today is also genotype, and genotype refers to the genetic makeup of alleles. So examples of genotypes would be two uppercase t's. That would be a genotype. An uppercase and a lowercase t, a genotype, and then also two lowercase t's would be a genotype. Okay? Now, what those represent, whether they're tall or short, they would represent what the phenotype is gonna be in the end. All right, so let's talk about the steps of setting up our cross. The first step, and this is the most important step, is you have to identify the parents properly. If you don't identify the parents correctly, then the rest of your steps is kind of a domino effect. They're not going to be correct. And so we got to pay attention to things like homozygous and heterozygous. We got to know what letters we're using. We got to know what our traits are, and those types of things when we do that. So as I said earlier, we are going to use T's and the traits tall and short. Tall being dominant, short being recessive. So capital T will stand for tall, lowercase t will stand for short. And then two, we have to do a process called segregation of the alleles. And basically segregation will show meiosis happening. You're going to go from a diploid cell to a haploid cell to show the creation of a gamete, like we talked about during meiosis. So let's start putting this into action. So my question for you today is, we want to cross a heterozygous tall parent with a heterozygous tall parent. So we're going to look at pea plants, is what we'd be doing, much like Gregor Mendel did. So both plants this time have the same trait, which is heterozygous tall. So I want you to think for a minute, what does heterozygous mean? Same or different? Okay, so it means different. So that means that in order to make my parent, I have to have two different letters. So in this case, I'm going to have an uppercase T and a lowercase t for each parent, each of those being heterozygous tall in this case. So heterozygous tall crossed with heterozygous tall. Okay, step number 2, we have to do segregation. Segregation is simply going to look like this. We're going to separate our letters apart from one another. So I'm going to take that T and kind of separate it away from the other T. And you'll notice I'm going to color code these so you can tell what we're going to be doing with the letters from here on out. We want to do the same thing to the other side, so we separate our two T's. And these circles on here are representing the gametes from these parents. So the parents you see are 2 letters, so that's diploid, and each of the gametes, the circles, are haploid, having only 1 letter in them. Alright, so that's the second step. In our third step, we're going to do the hardest thing of them all, and that's to draw a square and put a little cross inside of the square. And that's simply the Punnett square itself. So pretty easy step for the most part. Now we need to place the parents on the square, and there's really no rhyme or reason necessarily, but normally what I do is I take whatever parent is on the left and I put it on the side. So what I'm going to do is put my T there, so one gamete's represented there, and then the other T going down there. And then I'll take the other parent on the right hand side and put it across the top. So you'll see I put my green T where I do, and the brown T where it goes. So each T has its either row or column that it sits in inside of that box. Some worksheets and things will have these boxes. You'll have actually a square that actually might even go around the parent letters just to separate them so you can see that. But in this case, we're not going to do that. I don't think it is necessary. Alright, so I'm going to switch. I'm going to make a little larger Punnett square here. So what are we going to do next? We're going to kind of place our letters where they belong. And, there's a couple different ways you can do this, but simply what I'm going to show you is I'm going to take the blue T and I'm going to transfer it across to both boxes that it kind of touches. Okay? And then I'm going to take the green T and do the same thing. I'm going to move it down. And then I'm going to take the red T and I'm going to move it across. And then the brown T will move down as well. And so what you see, we're kind of taking these 2 letters and combining them. We're taking these 2 letters and combining them. These 2 here and combining them. And then this one and this one, combining it here. Now, a general rule of thumb, something I want you to pay attention to, is notice which letter I put first when we combine the big letter and the lowercase letter. I'll always put the uppercase letter first in a situation such as this. Okay, just a general rule. Just do it. Trust me, it'll save a lot of confusion down the road. Alright, so now we have our Punnett square. What's the next thing that we do? Well, the next thing is probably the most important to kind of finish the whole thing up, because we're going to come up with our ratios or our probabilities of how things happen. Now, there's some shortcuts and things to doing these ratios. However, I want you to learn the longer method, and this is what you're going to use in class. So no shortcuts, please. The first ratio we're going to figure out is going to be the genotypic ratio. And this is a ratio of all the different allele combinations. So if you guys remember back, genotype and phenotype. Genotype refers to the letter combinations. So in this case, we have 3 possible genotypes anytime you do a Punnett square that looks like this. You could have 2 uppercase letters, an uppercase and a lowercase letter, or 2 lowercase letters. Now, anytime you do a Punnett square like this, you have to have these 3 possibilities. Now, we don't have it in this situation, but there are times that you may not have any of a certain one, but you still need to include zeros on your ratios. And the reason why is because we will know where that 0 belongs to know what the combination possibilities really are. So simply, what I'm going to do is just go through and count how many of each I have. So I've got in my boxes, I notice that there's only one big T combination. So I'm going to write a 1 there. I got a big T, little t, big T, little t. I have 2 of these, and then 2 little t combinations. So I count them up. That's what I've got. And so I'm going to write this out in ratio form. And the ratio form that we use for Punnett squares is not fractional. It would be a ratio that looks like this. We would call this 1 to 2 to 1. Now, there is a specific order that this goes in. The first number is always how many big letter combinations we have. The second number is always going to be how many big little letter combinations we have. And the last one is going to be how many little letter combinations we have. So, once again, it's important if there's a 0 in there, you need to include it. So if we did this Punnett square and we counted 0 of these, 2 of these, and 2 of these, I would have to rewrite my ratio so it would be 0, 2 to 2. So please make sure that you include zeros in the ratios if they need to be there. Our next ratio we'll do is the phenotypic ratio, and this is going to be a ratio of the actual observable traits. So in this case we use tall and short as our traits. So now we go back up to our box and count how many are tall and how many are short. So we look here, it's got a big letter, so it has to be tall. There's a big letter involved here, so it has to show the dominant trait, which is tall. There's a big letter there, so it has to show that it's tall. And here I have no tall letters. All I have is 2 lowercase, so that's going to show the recessive trait, which is short. So if I do a little bit of counting, I have 1, 2, 3 tall, and I have 1 short. So when I write this ratio, I'll do it the same way. I'm going to write the ratio 3 to 1. The first number is going to be the dominant trait. The second number is going to be the recessive trait. So I have 3 to 1. Once again, if you have to count everything. So if you would count this and you had 4 tall and 0 short, you would have to write your ratio as 4 to 0. Also, this question always comes up, can I put zeros in my ratio? Yes, that's one rule we're kind of breaking in math. Normally you would not do. Also, if I counted this and I had 2 talls and 2 shorts, my ratio would be 2 to 2. Do you need to reduce it? The answer for this class is no, not at this time. So leave your ratios big, so 2 to 2 would be fine. I really don't want you to reduce it at this point of time. We want this to equal 4. And something to think about, this number plus this number plus this number equals 4. This number plus this number equals 4. And we know that because there's 4 possibilities in this box, so we have to have 4 as our outcome there. So just a little trick to think about. So my suggestion is to do a couple of examples, and these would be ones that you may want to do and then show your teacher, and they can kind of let you know whether or not you did them right. So one would be, here's the 2 parents. You have a heterozygous tall and a short parent crossed together. In this one, you've already got the parents, so now you can start with step 2. You really don't need to do step 1 now, and then go from there and show both types of ratios. My second example would be this one. Now you got to think a little bit more. I want you to cross a homozygous tall plant with a short plant. So now we got to think, and this would be definitely one. If you cannot work out number 2 on your own, see Mr. Wanamaker or any of your teachers and get help before you move on any further. So this is just kind of a quick tutorial on how to do a monohybrid Punnett square. Our next step will be to do a dihybrid Punnett square later.